Thanks, James, for that gravity-defying sharing. Continuing on the fight against climate change here, we have Michael Koepke to enlighten us on another way to reduce pollution by converting them into useful products on an industrial scale. Take it away, Michael.
Hi, thanks for tuning in. My name is Michael Koepke. I'm the VP of Synthetic Biology at Lanzatech. Lanzatech is a company that recycles carbon today for a cleaner tomorrow. We're a late-stage biotech startup founded 15 years ago in New Zealand and now headquartered in Chicago in the US. We raised over $400 million in capital over 5 equity rounds. We have about 200 staff and more than 750 granted patents. You may already agree that biology is amazing. If not, I can hopefully convince you over the next 25 minutes that biology can do things no other human-made technology or chemistry can do. It can operate at multiple scales, from nanoscale to global scale. On the top left, you can see a device called Flagella, which is a nanoscale motor smaller than any human device that we can manufacture today. On the other hand, on the top bottom left, you can see a algae bloom that's a truly global scale and really tiny organisms. The power of biology is that it can self-replicate and evolve complex function. And what's truly exciting is that it has a programmable genetic code. As humans, we're making use of biologies in thousands of years and across cultures. Think about alcohol fermentation like beer and wine. Start of the last century, however, we were also able to make chemicals using Bowel Manufacturing. One example is the Weizmann process for making acetone and butanol from starch. This process has been truly decisive in the world wars, for example, where acetone was required as core diet and was then produced via this process. Chaim Weizmann actually later on became the first president of Israel as an acknowledgement of his efforts. Fast forward 100 years, we've become really effective in biomanufacturing. Think about corn ethanol production. The last decades, as you can see here in this chart, we got really good at making this, and it's become a really mature technology. Biology also offers production that can be at smaller scale compared to chemical production, and it can be distributed, which I think is an exciting aspect. Nevertheless, there are challenges. One particular challenge is the feedstock that you have to grow. You can only do that in certain geographies. And of course, there's more and more demand on agricultural land with increasing population and at the same time climate change. This in turn leads then to fluctuating prices. So it's generally really hard if you have to make commodities from another commodity. One other challenge is that using sugars, you only have a yield of maximum 66% because you lose a third of the carbon to CO2. So what if we can actually make use of other feedstock and expand our options? Think about trash, which is filling up in our landfills. With the technology we have to date, only a very small fraction can be closed loop recycled, as you can see here in the chart. And we do see leakage into the oceans, so microplastics have become a huge problem, for example. Likewise, what about our emissions that end up as pollution in the atmosphere? CO2 levels are at the record highs since recording started. And we actually see unrest, especially with young people. Some actually already have looking at colonizing other planets. But again, if you want to kind of make use there of feedstocks, you have to use something like carbon dioxide. Biology, in fact, is really well equipped to use carbon oxide. In fact, life originated in a very reduced atmosphere consisting mostly of carbon oxide several billion years ago, long before oxygen was in the atmosphere. It's speculated that life started down at those hydrothermal vents in the deep sea. And if you compare the composition of those gases flowing through those hydrothermal vents, it's not so different that you would find today in a smokestack of a steel mill, for example, or if you gasify waste material. So it's believed that the oldest biochemical pathway on Earth is this Wood-Lungdahl pathway, which is found in acetogenic bacteria. So this pathway enables to fix CO2 into the central metabolite acetyl-CoA, which is the building block of life. So one molecule of CO2 gets reduced in this Wood-Lungdahl pathway to a methyl group and then fused with another molecule of CO or CO2. The energy for this process can come either from hydrogen, or it can solely come from carbon monoxide by splitting water in a biological water-gas shift reaction. So this water-gas shift reaction can also be obtained catalytically, but it needs to be really high pressures and high temperatures. Biology can do this at ambient pressures and temperatures through enzymes. At Lanzatech, we're using a successor of such an organism that has this Wood-Lungdahl pathway, which is still believed to be the most efficient CO2 fixation pathway and still conserved in anaerobic environments today. At Lanzatech, we're using this microbe called Clostridium autoaetonogenum, which was isolated from rabbit gut. And this organism is capable of utilizing carbon dioxide, carbon monoxide, and hydrogen and convert that to ethanol. And we've developed a fermentation process around this. This process can use directly industrial emissions as input, think, for example, a steel mill. But we can also use biomass or municipal solid waste as input that can be gasified in a mix of CO, CO2, and hydrogen called syngas. And we can also use CO2 directly as input using renewable electricity as power source through, for example, electrolysis. On the product side, ethanol can be used as fuel. But and the biomass of the process, so the bacterial cells, are a rich source of nutrients, of proteins. But we can also make chemicals in the process, either directly or by upgrading our existing products. And in turn, those chemicals can be at the end of the life be recycled and turned back into the process. And we can not only do this at small scale today, we can do this at large commercial scale today. 2 years ago, we started our first commercial unit and already produced over 16 million gallons of ethanol at this site. This site is connected to a steel mill and uses these steel mill gases as feedstock. By doing this, we could avoid 95,000 tons of CO2 produced. And we can operate this plant in continuous campaigns over months at a time, as you can see here, and do this in context of a highly fluctuating in-gas that changes composition within hours. This is truly an advantage of biology, that it can handle chaotic inputs, which is something the traditional catalysis struggles with. Just as an analogy, think about us driving a car. I think it's kind of automatic that we can handle those inputs. For the biology, I think the microbes can really handle fluctuating conditions as well and adapt and still produce a steady output. This is an example of a demonstration unit that we have on using unsorted municipal solid waste, as you can see on the left-hand side as an input. As you can see, this is an even higher fluctuating gas stream as for example, a steel mill. depending on what type of trash is any given day in your kind of landfill. But biology can still produce this very stable, steady output, as you can see on the right-hand side. So we're building out the technology at the moment. In addition to our first commercial unit, we have several other plants using different kinds of industrial emissions, whether it's steel, whether it's ferroalloy, or whether it's refinery off-gases. And we have projects in development using municipal solid waste, plastic waste, or agricultural waste. Several of these projects are already in the late stage of construction, as you can see here. And the first next generation of plants will go online shortly. On the product side, certainly ethanol and the biomass are our first products, but we're not stopping there. We've developed technology to upgrade the ethanol to molecules like jet or PET. And through synthetic biology, we have the opportunity to produce directly different chemical products. And I'll walk you through that in a couple of minutes. So in addition to ethanol being used as fuel, we've also introduced our ethanol into consumer products and showed that it's suitable for that. So you can now buy cleaning products that are based on alcohol produced from pollution, from CO2. And likewise, we've then developed technology, catalytic processes, together with partners to upgrade our ethanol. So we have shown technology that to dehydrate and oligomerize our ethanol to produce jet fuel and diesel fuel. At the same time, ethanol can also be converted to polyethylene or PET. In fact, we already had first transatlantic and trans-Pacific flights with fuel produced through our process from pollution with Virgin Atlantic and ANA. The jet fuel that we produced has actually exceptional qualities. I think it is more energy dense than conventional jet. And in particular, it also has a reduced level of sulfur and aromatics, which makes it burning cleaner. And this is something that we've just actually spun out into a new business called Lanzajet, with the aim of having by 2023, 100 million gallons per year production capacity established. On the other side, we've also turned our ethanol into PET, which is now used in cosmetic bottles made from industrial carbon emissions. It was in partnership with L'Oreal. However, through synthetic biology, we're also able to make new products directly in the fermentation process, which I think is truly exciting. So you can imagine we can directly go from pollution to new products in one single step. The key challenge is those organisms that can utilize those gases are very robust, which is great for a process, but they're very hard to genetically modify. And in fact, even 10 years ago, they were considered to be genetically inaccessible. In the past 10 years, however, we've developed a comprehensive synthetic biology platform for the organism that spans an array of design tools. It spans also advanced genetic tools. It spans to high-throughput screening methods and models of the organisms, including machine learning tools. One example is this world's first anaerobic bio foundry that we have established that is capable of fully automated generating and screening thousands of gas fermenting microbes. This system is housed in a completely anaerobic environment and can handle those gases like carbon monoxide, hydrogen, carbon dioxide, which are explosive gases. So I think this system has extensive safety features around that. As you can see, the system comprises of several fully integrated units, several robotic instruments, and also microfluidic systems. So while we're now able to generate thousands and tens of thousands of strains automatically, we wanted to push this even higher to enable a more rapid way that we can generate new products. Therefore, we've partnered with Northwestern University and developed a novel technique to prototype enzymes and pathways outside of the cell. So we can, through cell-free protein synthesis, generate enzymes in vitro, and then mix and match those enzymes to find the best combination of different enzyme variants and the best level of those enzyme variants that we can then replicate in the cell. Importantly, we find that this approach is very predictive. So the combinations that we find in vitro working well typically also work well in vivo. More importantly, the combinations that does not work well in vitro, we also find not working well in vivo. So we can essentially screen down very fast to only a number of relevant combinations that we have to test in vivo in the cells. So this enables us a order of magnitude faster and higher throughput in testings than we could do in vivo. To accommodate this really high throughput that we have been able to establish over the past years, I think we also had to develop computer-aided design tools, much like you find them in other industries. We did this in partnership with Teselagen and Penn State. And this BioCat software can automatically design thousands of DNA designs. Importantly, I think this whole system is powered by validated proprietary algorithms, for example, on the codon usage of the DNA, as well as libraries of new genetic parts like promoters that can control then how much of a given enzyme you will get. And these design tools then really seamlessly feed into our modeling pipeline. So we've developed metabolic models as well as reactor models that are all connected with each other and ultimately feed into economic models. So in the middle here, you can see actually the output of our metabolic model. So we can very precisely predict with a certain gas composition what output of ethanol we can get. But more importantly, also, if you have a new product, what's the yields of this product we can expect? What steps do we have to do to optimize that? And the whole system, now that we can generate so much data, is supported through machine learning to optimize the process. So to give you one example, I want to share with you how we developed a process from gas to acetone. So coming back to the Weissman process that I mentioned in the beginning, that was able to produce acetone in a mixed fermentation with butanol and other products from starches. So through work by Professor David Jones, some of those strains that were used in the early 19th century and then throughout parts of the later century in other operations, for example, in South Africa, we had access to strains from this process. With sequencing technology now available, we could actually pull out the genes of these organisms that are responsible for making acetone. Unfortunately, none of these strains could grow on gases alone. But we could pull out those genes and now implement into our organism with the advanced tools that we have. So with the prototyping platform and also with the combinatorial approach in vivo, with our Bio Foundry, we could quickly optimize flux through the pathway by 20 times. However, when we first introduced this pathway, we still saw very little acetone and mostly ethanol production, and also another byproduct that emerged, 3-hydroxybutyrate. With our modeling capabilities and then also with systems biology, we could identify targets and then quickly eliminate those other products. And we can produce acetone now at over 90% selectivity. So we've made over 40% improvement from the starting point there. And we've scaled up the process now to pilot scale. We've also completed an LCA analysis, which shows that we can save over 185% greenhouse gas emissions over the current process. So currently, acetone is produced exclusively from fossil fuel, and per kilogram of acetone, you release about 2.5 kilograms of CO2. Whereas through our process, we can lock in the CO2 into the product. So we actually have a negative footprint there. So, yeah, ethanol is certainly our first product that we have in commercial pipeline. So we have product molecules like acetone, like isopropanol, and other molecules in the pipeline that we've already piloted today that will feed into this pipeline. And I think that the power of biology is really, we can just simply swap the microbe, use the same plant, use the same hardware, and make a new product. So I think this is truly powerful and a paradigm shift over the chemical industry, where you have a plant dedicated to one product. And it becomes really disruptive because, as you all know, I think chemical prices fluctuate a lot, and we can really uncouple this process, since our feedstock is not a commodity, and we can quickly react to changing prices. As I mentioned, I think we have developed processes for acetone, isopropanol, also butanediol. At lab scale, we've also demonstrated, for example, direct production of monoethylene glycol that we're optimizing at the moment. Also a range of other molecules, even complex molecules that have multi-carbon chains we can build from those one-carbon molecules. And it spans a range of different molecules, starting from different kinds of acids, different kinds of alcohols, diols, aromatic molecules, also esters, dienes, ketones, and terpenes. So we've demonstrated proof of concept for over 100 molecules that can be directly produced. But from our modeling pipeline, I think we know that it's feasible to produce many more molecules as well. So with this, I think what we're really aiming for is enabling a circular model, where we can capture and use waste streams like and pollution, and produce either fuels or use chemicals that can go into carbon smart products that eventually at the end of the life can be recycled and brought back through the process as well. And much like that, I think replicating what we find in nature. In nature, there is no waste. If you consider, for example, a cherry tree that produces thousands of blossom, which then creates food for birds, humans, and other animals to grow on the effort of this one tree. Before I close, I think one point I wanted to make as well is I think as a field, I think we must really adopt technology neutral positions and support all solutions. I think this problem is massive. I think we need a lot of different technologies to access it. And we must quickly fail and move on to identify technologies that work. I think it's critical to collaborate and address also environmental concerns and get fuels and chemicals to the market quickly. I think it's very difficult to scale up and very costly to scale up first of its kind technologies. I think this is really an area which I think is quite important. So I think through policies, definitions, and funding, we need to drive the technology, because it can either drive or block the technology, and we need to act quickly. I think the Earth is really on fire. There's not much time left to combat climate change. with that, I think I want to thank you for your attention, and especially thanks to the organizer for the opportunity to be here. And no carbon left behind.